I'm going to move on to Petra now on module number 2 and hand it over, as she takes over on gene therapy development considerations. Petra, over to you.
Yeah, good afternoon again, and thanks for having joined the webinar. So as an outline, if I could have the next slide, please. I seem to have lost the ability to advance. Thank you. I would like to review what gene therapy actually is and how it could be applied to Duchenne muscular dystrophy. Also, why do we think this is a really exciting moment for gene therapy, and what are the challenges on the other hand? And then finally, I will end with reviewing what we think might help with the successful development of gene therapy. So getting to the first point here, what is gene therapy? So gene therapy, if you look to the left here, is essentially a way to correct defective genes that are responsible for disease. So we can introduce a new gene that would be a functional replacement. We could use a modified version of a virus to transfer that gene, and the virus itself, importantly, does not cause disease and is not passed on to others. So when you hear virus, you think cold and that it causes disease, but this is different. We just use the virus as a delivery truck, as a vehicle to bring that modified gene in, but the virus in itself doesn't cause a disease. So you see here on the right side a schematic representation with the vector here. This is like this shape with a DNA symbol inside, which means that the vehicle there, the shell, brings that new DNA into the cell. And if I can go to the next step, you can then see here how it gets into the cell, or event right to 3, sorry. So basically, thank you, the shell breaks down and releases that genetic material that can replace the missing gene or the dysfunctional gene. And then third step, please, what happens next is that the new gene is in place and the protein that is required to help the patient is actually being produced. And now let me step back a little bit, and many of you remember this from biology. So how does this actually work? So there you will hear these words: transduction, transcription, translation. What does that mean? So transduction is when you see again this virus, this we call a vector, the delivery truck with the DNA inside, getting to the cell. When it gets in there, that's what we call transduction. So the delivery of that gene, the transgene, the new gene that's being introduced into the cell, that's called transduction. Then the next step is transcription, where we have to get messenger RNA produced. And you see here how the DNA has a code, a letter code, and I'll talk about that a little bit more. That is essentially like the script and the recipe book. And then from that, the mRNA, the messenger RNA goes out, it's produced, goes out, and that's when translation occurs, when the mRNA is made into a protein, when the mRNA guides how the proteins are put together, protein synthesis. So these are important concepts to remember when thinking about gene therapy. And so now I told you just now about the about the genetic code there, sort of the cookbook or that the script that determines what proteins are made. So if you look at this in a different way, you see on top here these the DNA, and it's really important to remember that the DNA has 3-letter codons. So meaning that there's 3-letter words that basically can be read, made into this messenger, can be read, and they determine what kind of protein is put together. You see here the different amino acids that are put together to make a protein, like dystrophin. And the 3-letter words determine what kind of a protein is being made. So now let's look at this in the context of Duchenne. How could some of these gene therapy approaches work? And this is just to illustrate this. Of course, these words are not really what is there, but just this as an example shows you a sentence that is made out of all 3-letter words. And you can obviously read it. You can read the mad cat ate the fat rat and the big cat. So don't worry about what it says. It doesn't make any sense. But just to show you now what happens when 2 letters are missing. So you can read the mat, cat, ate, the, and then you kind of get stuck because when you go every 3 letters, this is all, you can't understand it anymore. And that's basically when the protein production stops because the message is not clear anymore. The body, the cell doesn't know anymore what kind of protein to make there. And you get, in the case of Duchenne, you get a very short piece of dystrophin that doesn't really do the job at keeping the muscles healthy. So when you've heard about exon skipping, what that does is that it basically skips over a number of letters to get back in frame, to get back into a message that you can read. And here the sentence is shorter than it's supposed to be, but it's still a good sentence. So there is still some function left here. So the cell machinery can at least continue reading through the rest of the transcript, and we get a shortened form of dystrophin, but at least we get some dystrophin. And that is kind of the general principle in a schematic way that is behind some of these exon skipping approaches here. Now let's go to the next approach, read through. What does that mean? So you have again the three-letter words here in the sentence. And if you have what we call a nonsense mutation, meaning that there is by mistake a stop sign introduced, so some of these 3-letter words mean stop to the body, then you basically can't go beyond. Again, you get a short piece of gene, short protein that doesn't really function well. So then there's different medications that can read through that stop codon. So they have enough, give the machinery enough power to read through. Now this, of course, would only work specifically for people who have that stop codon, similar to the exon skipping that works only on people who miss that particular message there, right? But similar to exon skipping, it uses a small molecule often, and it can push through there and result in the production of some dystrophin protein. Now we're coming to gene replacement. So here again, and you know this by now already, you have the kind of sentence that is disrupted and becomes basically unintelligible, and you could have any kind of mutation, but it doesn't matter which one, because what happens here is there is this vector that you've seen earlier that brings just this new piece of DNA in. And in the case of Duchenne, as you may know, the issue is that what we need to bring in is very large, so that some of these approaches bring in a smaller piece in the hope that that could help enough to make patients feel better and function better. But it's unfortunately, with some of the viruses like this AAV virus, not possible to put the entire message, the correct message, inside the virus. It just doesn't have enough packaging capacity, if you wish. So the virus it doesn't cause disease, and it can bring this shortened version. It's currently being tested in clinical trials. That's gene replacement therapy. And now we get to the last approach here that I want to briefly review for you, which is gene editing. So again, you have all kinds of mutations, deletions, and in the case of gene editing, basically, it is a very targeted change that is made. It's almost like a repair that's made with enzymes that can cut into the patient's DNA and remove or replace. So this is really, something that is not in clinical trials at this time for Duchenne, but that, is basically another potential approach. And it could also be combined with a vector approach and could be delivered with a viral vector. So this is just sort of setting the stage. And now let's think about why this is really an exciting moment for gene therapy. So the history of gene therapy is rather long, actually, not in the big picture of medicine, but it's been going on for over 20 years. And there were a lot of advances that were needed. And then there were also some setbacks. So we have to understand that gene therapy is still new and is certainly something that has concerns with regards to safety. And sadly, in the 1990s, a patient died from gene therapy, which led to a lot of extra work that was done to ensure that gene therapy can be as safe as possible. And there's a little bit of a gap perhaps then, but just in the last couple of years, we have really seen breakthroughs that I think make this very exciting. The FDA has approved a gene therapy, a cell-based gene therapy for leukemia, and then there has been a gene therapy approved that is given into the in vivo, meaning into the patient, not just into the cells outside of the body, for inherited blindness, and then also earlier this year for spinal muscular atrophy. This is why this is really exciting to see that finally gene therapy made it all the way to approval. And I mentioned earlier this particular virus that's called adeno-associated virus. And this one has been used in some of these gene therapies that I was showing you, the one for blindness and for SMA, because it can reach many different cells, many different organs and tissues, including muscle. It has different types so that it could be somewhat targeting certain cells more than others. It doesn't cause disease, and it's not known to integrate into the patient's genome, so that is thought to be safer, but it does clearly elicit an immune response that you can imagine. If you get a virus into your body, that your body has to react. And this one, however, is thought to be somewhat less maybe than previous viruses that were used. And it can also be modified in a way based on what kind of type is used and also what kind of promoter is used. That's like the switch, if you wish, to be a little bit adapted to different applications. So what are some of the challenges in gene therapy? Well, one of the challenges is getting into the right place safely. And the way that this can be optimized is by picking what, as I mentioned earlier, from these AAV types, for example, a particular type that goes more to some cells or tissues than others. Then there is the switch that I mentioned, the promoter. So researchers can pick one that is on all the time, or that it only kind of tries to target specific cells where it turns itself on. The route of delivery is very important. So in Duchenne, it would be intravenous, or it may be even more local, but in other diseases like the blindness, it's into the eye, or in diseases of the nervous system of the brain, it could be into the spinal fluid. And you see that as an example here on the right. So we use testing in mice or in monkeys to see where the vector goes. But in clinical trials, you can then look at maybe patient biopsies to see if it's expressed, if you can actually see some of that dystrophin being made. And an important other part of getting to the targeted cells is antibodies. So our bodies have antibodies against viruses, and these viruses are naturally occurring. So that's another issue we have to look for, if patients have already antibodies, and how to help them still maybe get access to gene therapy. And also we want to look at the antibodies that are produced as a result of being exposed to this virus. And safety is very, very important. Laboratory studies in animals have to be performed to understand the risks and benefits, and that's critical. And then once patients are in clinical trials, they must be very carefully monitored, and it's probably burdensome for patients to come to many study visits or get many blood tests, but it is very important so that these clearly, evident safety risks are properly monitored for and prevented or mitigated to the extent possible. And the adverse effects that we know about include, to some degree, responses of the body of the patient to the virus that is given, to this treatment, to the vector. And this can present in many different ways, but clearly one way we know about is, for example, increased liver enzymes, which can be monitored and have to be monitored in patients who are in clinical trials. There are some drugs like steroids that you may have heard about that are given to patients who get gene therapy to prevent or help with these adverse effects, but it's not certainly 100%, and there are still adverse effects. And in addition to the known ones, there may also be unknown effects and risks. So it's a new therapy, great potential, but we have to keep in mind that there is also risks and uncertainties, of course. And another challenge for gene therapy is the scalable manufacturing. So then if a gene therapy can be developed, and if it can get through clinical trials and even get through approval, how can we make enough and ensure that it's pure and safe and robust? And the way gene therapy is produced is that the vector here has to be put together into cells with some helper plasmids. That means because the vector itself is engineered, it can't really replicate anymore. It needs some helper plasmids there. Then it's put into these cells. They're called HEC293 cells in this case, that's human embryonic kidney cells. And it's basically incubated, and these cells produce the virus, basically. And then it has to, however, the cell becomes sort of the machinery for this. And then the virus that is then removed from the cells after they have done their work has to be purified. And this is a process that is relatively new, and a lot of people work on evolving it and making it better, but then there is massive scale-up needed in order to have enough for clinical trials, and certainly then afterwards for all the patients who need it and could benefit from it. So I was asked to comment on what some of the success factors may have been for these recent gene therapy approvals. And I think it starts really with collaboration. I think it starts with the patients and the families, with people who, really altruistically participate in natural history studies, in clinical trials, and studies on the patient perspective. Without that, I think we would not be where we are now in some rare diseases, neuromuscular diseases. So, thanks to everybody who has done this, and people have often participated in studies when there was no treatment, but it still helps and gets us where we are now. So strong patient groups are very important, who can provide education, information on trials. And when we think about planning a new trial, the more information we have, the better it is, and the faster we can make progress together. Clinical trial readiness is really what that boils down to. It's having good data, outcome measures, and a good community that works together so that we can test these new therapies that we hope there will be more coming. And finally, for gene therapy, it's also important that we think that it works better early. And so early diagnosis is important. So awareness among pediatricians, among neurologists, so that they can find out early on what kind of mutation or what kind of problem a patient exactly has who could benefit from these new therapies. So I hope this was a helpful review of gene therapy and some of the challenges, but also opportunities. And I thank you for listening this afternoon.
Petra, thank you so much. Sharon, you back.
I am. Sorry, I was disconnected and I'm back.
No worries. You can have it from here.
Great, thank you. So a lot of questions have come in during Dr. Kaufmann's talk, and one question that I think is very interesting is in gene therapy trials for Duchenne, you often hear readouts where investigators talk about dystrophin intensity. And the question is, what exactly is meant by dystrophin intensity? What is that reporting specifically?
Yeah, I think this it goes back to what Dr. Marks explained, where people often try to look at a biomarker. So, of course, in the end, the goal is to help people function better, to have greater muscle strength and feel better, but that sometimes takes longer to measure, and it takes larger trials. So what people do then is they look at the dystrophin itself, and they do muscle biopsies, and there are different laboratory tests that one can do to try to see. So basically what you try to do is when you have a patient with Duchenne muscular dystrophy, and you look for the dystrophin, either with basically immunohistochemistry, where you see it under the microscope and then we make it fluorescent so you can see it, or you could use something called a Western blot to quantify it, to measure how much you have. So basically these lab tests can measure it, and when people talk about the intensity, they look at basically what you get before. They try to compare before treatment, where you don't get much, and after treatment. So you look at how much dystrophin there is and how it's distributed.
Okay, and there's several questions here about different types of mutations, whether stops or duplications, and how well gene therapy would work to address these different mutations.
Mm-hmm. So as you saw earlier, some of these approaches are targeting just specific things like a deletion or a stop codon. So gene therapy in principle could work for different kinds of mutations because it would replace some of the missing gene. The issue in Duchenne is, as I pointed out, that in some of these, the vectors we have right now, is, for example, the AAV vector, can only deliver a smaller piece of DNA, much smaller than the dystrophin gene. And therefore, it cannot deliver the fully functional gene that makes the fully functional protein. And the expectation, therefore, is that it could help, the hope is certainly it would make patients better, but it would not be a full kind of functional gene. So maybe going from, people say often from a Duchenne muscular dystrophy or a more severe muscular dystrophy to something milder, more like a Becker dystrophy. That's, I think, what people could expect.
Okay, and then just one more quick question, I think, before we move to the next module, and that is a question that was written in advance. And it was about whether or not gene editing and gene therapy could work together in a treatment approach.
Oh, that's a challenging question. So the hope, of course, that some of these new treatment modalities will very soon, help patients, with Duchenne. I mentioned in the slide on gene editing that the gene editing could be potentially delivered with a vector. So I don't know if that's where the question is going, but I'm thinking maybe people are asking if one could combine the different treatments. And that right now, I think we have to go one step at a time and look at them individually, and then let's say once they are shown to work individually, then as a future step, one could think about potentially ways how they could be combined. But certainly, that is also the case. The question was about combining, different innovative gene therapy approaches. You could also, of course, think that somebody could get gene therapy and still get some of the maybe steroids or certainly all kinds of physical therapy and other things together at the same time. So that's all things that would be considered once one of these innovative treatments has been approved and shown to work.